discussion is the principles of inheritance and variation. And one of the pictures shows a child genetically linked to his parents by DNA, and the other picture depicts a random cross, about which you'll be learning soon in my lectures. So guys, stay tuned. Now, let me move through some important terms for better understanding. Heredity, a very important term, as you know, is the transfer of characteristics from the parents to the offspring. And you see the picture of the family, we see the children resembling their parents and also the grandparents. Yeah, it's because of what we call inheritance. But we also see that every member in the family differ from one another in some way. So what I mean is, the degree of difference we see is termed as variation, which technically is known to be caused from recombination during crossover. I think you would have noticed while learning meiosis in your lower grades. Also that other environmental factors can also have effect over it, which will be emphasized soon after we learn about the very basic unit of inheritance, which is called gene. And as you see in the picture, it's located on the chromosome. That is, it's just a part of DNA which is present in the nucleus of a cell. And here, we definitely have to thank the father of genetics, who is none other than Mendel, who worked for many years on garden pea plant and made really amazing observations. Also, I definitely recommend you to read the life history of Mendel, which will seriously drive you into further research in this wonderful field he worked on. However, moving into the topic, he observed 7 distinguished characters in garden pea plant, which are listed here, namely the form of seed which can be round or wrinkled. As specified in the slide, the notation goes as per the variant of the character, which is dominant over the other. So in the first trait, that is seed form, round is the dominant form, and so there is a capital R for round and a small r for wrinkled. And this applies for the other traits as also listed. And now we advance forward in learning other important terms. As said, genes, the unit of inheritance, is just a part of DNA in the chromosome present in the cell's nucleus. And alleles are the variants of the same gene. For example, consider a gene for the flower color, and you observe for a set of homologous chromosomes, the allele for purple and white flowers are in their respective loci. And imagine one of the homologous chromosomes has the allele for purple flowers, and in case the other chromosome also bears the same allele, then it's termed as homozygous, that is, both purple flowers. And if one bears purple flower allele and the other one has white flower allele, then they are heterozygous chromosomes. And now, let's try to understand about the terms genotype and phenotype. Look at the picture below and you can see 2 variants for the trait hairline of a human being. One is a widow's peak and the other a straight hairline without any peaks. Now, you find so many W's on screen, which actually is the notation for the gene hairline, and since widow's peak is dominant, that is, it overrides the effect of the straight hairline allele, which is a variant of the gene hairline present on the other copy of the chromosome. And so, the person who expresses a phenotype, so basically phenotype is the appearance when viewed physically. So if the person expresses a phenotype of a widow's peak, then the possible genotypes could be capital W or capital W small w. Since small w does not get expressed when present along with capital W, it's sort of treated as a recessive trait. And the genotype of the person having a phenotype of straight hairline has to be only small w. And as legends say, the body behaves on what the DNA says. Now, moving a bit deeper, let's start learning about inheritance of one gene. Mendel actually performed crosses among several pea plants and gave his observations. At first, he selected true breeds. Okay, so what are true breeds? True breeds are the plants which show purity in the trait they possess. More technically, if a dwarf plant possesses a small t and a tall plant possesses a capital T genetic composition, then they are said to be true breeds, which are believed to be generated by self-pollination method. So now, Mendel crossed a true breeding tall and a true breeding dwarf plant, and the first hybrid generation, also called the first filial generation offsprings, resembled only one of the parents. That is, they were all tall phenotype, and please be very careful while deciding their genotypes, as here every F1 offspring is heterozygous tall. And by heterozygous tall, I mean the genotype capital T and small t. And when any two of the F1 offsprings were selfed, the second filial generation showed both parental traits but in different proportions. As you can see, the dominant trait here, the tall one, is mostly seen than the dwarf offsprings in F2. Very importantly, we see no blending of variance here. That is, there is no plant of height intermediate between the tall and the dwarf. And you may now be like, oh my god, maths in bio, I'm done. But hey, actually, to make the job easier, that is, for any genetic cross, we can determine the possible genotype of the offspring by a very useful method, which is the Punnett square. It's just a table to be filled prior with the data needed to be matched. For example, while finding the F1 offsprings, we write the male plant's genotype or the alleles on the top and the others in the left vertically, and simply we combine the letters and fill the boxes, and finally we obtain the genotypes of the F1 offspring very easily. Similarly, continue doing for the F2 generation also, and while carefully noticing their genotypic ratios, we see that the plants whose phenotypes are same might be genotypically different. That is, it can be homozygous or heterozygous. And after counting from the Punnett square, we get the genotypic ratio as 1:2:1, which typically means 1 capital T, that is homozygous tall, 2 capital T small t's, that is heterozygous tall, and 1 small t, that is homozygous dwarf. And the phenotypic ratio, very simply based on physical appearance, it's just 3:1. I mean 3 tall and 1 dwarf. And now, we're slowly moving to another interesting topic, the test cross. And guys, don't worry, it doesn't involve much math. So yeah, definitions apart, I would like to explain practically what a test cross is. So for example, let's consider in your garden, there's a flower whose central part is yellow, and assume it to be the dominant trait, and its alleles are obviously capital Y. And for the one with white central part, it's obviously small y. So now, when you go to your garden and just randomly pick a flower with the yellow central part, but you're totally confused about its genotype, as there are two chances. One is capital Y, and the other is capital Y, small y. To know to which one of the two categories the flower you picked actually belongs, we're going to do something which is called the test cross, which is actually a cross between a homozygous recessive, here the white-centered flower, with a genotype small y, and the flower you bought from the garden. So after the cross, in case you observe all the offsprings having the yellow center, then you have plucked a homozygously dominant flower with the genotype of capital Y. In case you could also find 50% of white-centered flowers in the offsprings, then you had plucked the heterozygously dominant flower of composition capital Y, small y. Please have a look on the Punnett square for more clarity. So based on all the previous observations, Mendel stated the following laws relating to monohybrid cross, that is, cross involving only one trait. And the laws are: law of dominance, which states that one of the alleles is dominant and gets expressed in the phenotype in case of the heterozygote. Example, when we cross homozygously tall, which is capital T, and dwarf small t plants, in the offsprings, we get all the tall plants having the genotype of capital T small t. So, tallness is a dominant trait over dwarfness. And the other law is law of segregation, which states, as each allele separates during meiosis at the time of gamete formation, there is no blending and characters are passed to different gametes. Homozygotes produce only one kind of gametes, which is capital T, and heterozygotes produce different kinds of gametes, which can be capital T or small t. And now we come to the last topic of my current lecture, which is incomplete dominance. Have you ever noticed some of your hairs might be full of curls, let's say C1 genotype, while some with full straight hairs with C2 genotype, and some have a blend of straight and curl hairs, which we call wavy, maybe of C1 genotype. Not only in hair texture, even in hair colors, you might have observed the blending of parental traits. Here, you can't find out which hair type is dominant, as you can see blending of different hair textures, and that's why we write the notation for both the alleles in capitals with subscripts to differentiate one another. Another example would be the flowers, which are red and white in color, and when crossed, they yield F1 offsprings of pink color of genotype R smaller, which is actually a blend of the true breeding red of genotype R and true breeding white, which is smaller. And to find the F2 hybrids, let's solve by Punnett square, which gives the data as 1 red homozygous, 2 pink heterozygous, and 1 white homozygous. And I hope I've made my points clear. If you have any doubts, feel free to put in the comment section. And hereby I complete the first part of the lecture Principles of Inheritance and Variation. And I'll be continuing with the topic Codominance in my upcoming lectures of the same unit. So stay tuned guys, and yeah, thank you.